tardigrades, definitely not funguses. So, and I'm at Baker University in Baldwin City, Kansas. Got my education through Montana and University of New England in Armidale, Australia, over the years. And I'm going to talk about the critters in many different ways. One of the most famous space flight and things like that. They did appear on the Cosmos series with Dr. Tyson. And we discovered at least 5 major errors they made in presenting the animal. And when we talked with the animators, they told us they were not allowed to do fact checking. Anyway, you can find them anywhere. Here they were discovered in a Japanese parking lot. They have unique proteins to handle this desiccation factor, which we'll get to in a little bit. Okay. DARPA is using them to search for life, potentially on other planets, and to protect life here. And they putting some big bucks into some of this research. In the Antarctic, a couple of years ago, they dredged up something from one of the deep holes they were drilling 3,000 feet into the ice, and they pulled up a thing they thought was a tardigrade, and the picture on the right there is what they pulled up. Within 24 hours, I had the pictures and had the specimens on the way, and it's a piece of dirt. But they made national and international news with it, so everybody was happy, except me. Some recent work in Japan has taken a sample out of a refrigerator that had been frozen for 30 years and found live active animals, which is sort of an underplayed aspect. The 30 years is pretty spectacular, but what they don't say is that these animals were frozen when they were active, not when they were cryptobiotic and desiccated. So it's a second avenue of survival, well worth investigating. We found them in the tops of trees, and we've done a REU project here with students, some of who were disabled, and we had them climbing 100 feet into the tops of trees and participating in our research projects. Okay. Tardigrades are movie stars. You may have seen them in Ant-Man and the Wasp. They're TV stars. The navigator on Star Trek Discovery is a tardigrade. Yeah. There's music with tardigrade songs in thing. I don't know why they put a pelican on the record jacket cover, but they did. Okay. There's some beautiful images out there of tardigrades. This one is a Ramazoutius from a nature system. And then University of Baltimore produced this one. Within hours of this being shared, it was already stolen, and people were making T-shirts and what have you out of it. It's gorgeous. They sent it to me to see if I could identify it as a Hypsibius. Yeah. Tardigrades can be used in political satire. Regardless of where you are, the similarity is unmistakable. And in real literature, Dr. Wilson is a friend, or was a friend, and mentions tardigrades in almost all of his books. We never got to do a project together, but it was a real delight to work with. Okay. American Museum of Natural History has done a Life at the Limits study in which they have 6-foot-long tardigrades that they hang from the ceiling for public presentation. Yeah. There are some minor museums. This is our museum at the Baker Wetlands here in Kansas, where we describe the new species that we found right on our own property. Okay. Popular journals, American Scientist, I've managed to get a paper approved there at once. And they've been shot from guns. They've survived half the impact of a 9mm round. And they've been on the International Space Station. Okay. They've been found in Dominican amber, 16 million years old. And we actually have some other amber specimens that are back closer to 75 million years. Their body shape hasn't changed much over time. They're probably very slow evolving. So what is a tardigrade? It's a microscopic animal that lives in mosses and lichens. It looks like a little caterpillar, and it's less than 0.5 millimeter in length. I tell people it's the size of the period at the end of a sentence. It can be seen, but the light has to be just right. Okay? They have this spectacular appearance, and they have claws, they have faces, they have their parts. They're a pretty spectacular looking animal. This one I love. And the face that's there on the mouth is actually an artifact of folding. It's not a real face. We don't have visible eyes on this particular guy. Yeah. So it is a phylum. It has 5 body segments, separate sexes, and lays eggs. It has 4 pair of legs, each ending in a set of claws. It has a nervous system with light-sensitive eyes, but they don't erupt to the surface. It has a full digestive and excretory system, complex mouth and pharynx structures. It's well-developed muscles, periodically sheds its cuticle, but it has no respiratory, no circulatory, or a skeletal system. Those are the complex of characteristics that make it a unique phylum. In cross-section, it looks much more like an animal. It has a dorsal brain and a ventral nervous system, and the nervous system is a double ladder with a major ganglia, 2 major ganglia in each body segment connected. Makes it rather both polycomorphic and apomorphic at the same time. The other really unique characteristic is that it only has a single gonad. Unlike almost all other biological things where they occur in pairs and are symmetric, this has only a single gonad, ovaries or testes. There we go. Tardigrades are famous for cryptobiosis, the art of desiccating with the environment and then surviving extreme conditions. But there are other phases, other stages of life. The active stage, of course, where it can eat, grow, move, reproduce, watch MTV, do whatever it wants, is in the middle. If there is not enough oxygen, it goes into asphyxia. And in this case, it bloats up like the Pillsbury Doughboy and floats around until the oxygen or the water level declines and it recovers to the active stage. If it is an aquatic tardigrade, which lives underwater all the time, it can form a cyst when the stream dries up. But for the most part, the terrestrial animals that we know about do cryptobiosis. They can react to freezing, they can act to a loss of water, or they can react to an increase in salinity. So a variety of ways that they desiccate to become what we call a ton. In cryptobiosis, they've survived in the laboratory 0.05 degrees Kelvin for almost a whole day. They've been exposed to and survived -200 degrees centigrade for almost 2 years. They've survived plus 120, 150 degrees, well above the boiling point. 6,000 atmospheres of pressure, which is what you call stress, and still been alive, returned to active life, okay? Pure vacuums, well, impure vacuums too. Excessive concentrations of most common gases do not seem to affect them when they're cryptobiotic, okay? X-ray and UV radiation at the 13, 14 times what you and I can stand, right up there with cockroaches, and they still recover to active life. And they've been recorded to exist in this condition for 125 years, which is just absolutely amazing. That, of course, is the argument that they're capable of space travel. They can survive the heat, cold, time vacuum with little effect on the existing form. Okay. In 2007, tardigrades became the first multiple-celled animal to be exposed to space and return to Earth alive, and then grow and reproduce. True survival. Actually, they sent them up on the European space system, and they took the lid off the jar and exposed them directly to space, the sun, the radiation, the cold, the heat, everything, for a number of days. And they did several experiments and then brought them back. Did everyone survive? No. But about 75 to 80% of the individuals sent up returned to active life. Okay. And in 2019, we accidentally sent them to the moon, where they crash landed, and they're still there. NASA has refused to send a rescue mission. Not that they could. In discussing the possibility of space, Alpha Centauri is only 4.5 light years away. If tardigrades could hitchhike at 1/10 the speed of light, it would take 45 years to get there. At 1/25 the speed of light, it would take 112 years. So this is well within the documented survivability of the animal to the rigors of the trip. So theoretically, it's possible for tardigrades to travel in space. So where are they? Well, they're right in the middle of the animal kingdom, the life kingdom. And you can see very quickly they're ecdysozoa, protostomata. They're on the line with arthropods and above the nematodes. And what this doesn't really show us is that they are very possibly the first legged animals, and may have been the first legged animals to actually crawl out of the ocean. Worms and other things have wiggled around and surely colonized. We know that the plants and things have gotten out of the ocean, but this may have been the first one to actually get up and walk out. So we're kind of pleased with it. Classification. We have 3 classes, 5 orders, 100 families, But it's even simpler than that, because the middle Mesotardigrade, a class no one alive has ever seen. It was only reported once, and there's no specimens. Multiple expeditions to the Warm Springs in Japan have never produced the animal or anything even close to it again. So it's dubious in our records. So we have heterotardigrades, eutardigrades. Heterotardigrades are mostly marine, except for one family that we find on land. And eutardigrades are mostly land-based, with no exceptions found in the marine environment. Heterotardigrades are the ones with the plates, the spectacular spines and big claws, where eutardigrades are rather bland. They're nude, naked, smooth cuticles for the most part. Okay. Eutardigrades do take a number of shapes, some very elongated, some very cigar-like, etc. While the heterotardigrades are much more tank-like, little armadillo-looking organisms with dorsal plates and structures on their back, and all sorts of wild bumps on the cuticle. All of them have spectacular claws. When you have to look at them down here, the echinozoides, this particular specimen has 8 parallel claws, which would make a grizzly bear jealous. And there's been up to 13 in some species of this. Some are really fine tuned and almost looks like they'd be broken off. Some exist in pairs, others in parallel. There's a few singles, but for the most part, we use claws as a major taxonomic character. We use mouth parts. The animal is almost always transparent to some degree, and we can look at the mouth structure with a mouth at the front end and a buccal tube or throat down the middle, a set of what we call placoids, structures behind that in a muscular pharynx. These are actually the teeth. This is what it uses to chew, what it brings in. Some of the pharyngeal tubes are extremely narrow, only allowing the passage of bacteria or single-celled plant structures to get down to the pharynx. Others are quite wide. Off to the side, you see what are called stylets. These are spears. For the animal to eat, it walks up to a plant or another animal and compresses muscles between the stylet and the mouth, shooting a spear out of the mouth and penetrating the victim, the food source. Then it takes the pharynx and expands that, creating a vacuum and sucks in the juices. So can be pretty vicious for a cute little animal. Eggs can be very spectacular. Many animals lay the eggs inside their cuticle and they're just smooth and round. Others lay them free in the environment and they have these pretty wild projections for holding on. They look like little Christmas tree ornaments at about 80 microns in size. Tardigrades around the world, we've now got a little over 1,400 documented species. Not very many groups compared to many other organisms. In North America, we have about 300, a little over 200 in the United States. And since I'm in Kansas, we care about that. And this slide was prepared for a talk in Texas, so I had to put how many were on there. In North America, we've described 54 new species and 16 marine species. When you look at their continental distribution, the number of animals by state is rather discouraging. The maximums are 70, and there's only 3 states that have more than 50 species recorded from them, where Greenland and parts of Canada have much greater numbers. Europe and South America have far numbers. And this is simply a result of us not looking, okay? And we've got 4 states that have absolutely no records and 11 states with less than 5 records or species. Really discouraging when you're trying to do distributional and associational analysis of it. When we look at an actual GPS map of all the recorded records of occurrence in the United States, it's easy to see where the concentrations of data comes from. And I'm lucky enough to say that the one in New Jersey is one of my students. Montana is my master's. The stuff around Kansas City, of course, is our current project work. So we participated in the expansion of knowledge here. But look at the mass amount of space to which we do not know what species exist there. Tardigrades can live in moss, lichen, soil, leaf litter, beach sand, marine sediments, freshwater algae, up in the canopy we've discovered, and on all continents. We often find them in a variety of places. The neat thing about headstones in cemeteries is it gives us a starting date. Most other wild places, we have no idea how long the habitat's been there. But cemeteries have that unique dating possibility to it. Yeah, they can be moved around the world by birds. floating on plants, but wind is considered the primary mover of the animal. But we've been finding more and more and more alternative vectors to it. One of my students recently documented tardigrades on the actual feathers of birds and supports the migration between North and South America. I support tardigrade rain where they are blown around by the wind. The cryptobiotic animal is like a flake of dust, and it's easily picked up and blown between continents. It's the only explanation for the animals being found on remote desert islands and that sort of thing. Okay, it's one of the simplest animals to process. You just go out and take a sample. We use the handful as the standard collecting measurement. We drop it in a paper bag, which you get at the grocery store for 10 cents a piece for school lunch bags. We write all our data on the bag, wrap it up, and stick it in the closet. When we're ready, we take a sample out. We measure out a gram or so at a time and soak it for 24 hours. Examine it under a dissecting scope. Catch the animals and either preserve them in formalin or alcohol or mount them directly on a slide. Under the slide, we look at them at 400 to 1000x and try and identify them. We've been lucky enough to have described 19 species in 2 genera with 11 students, and we've got a half more sitting on our desk to get around to, which is really a lot of fun. My personal research has been Antarctica. My PhD thesis at University of New England, Armidale, Australia, defined the Australian Antarctic. We had about 28,000 specimens. It was a pretty big project. Okay. Tardigrades are a fantastic tool for meeting people. We became friends with Ed Wilson. One of my students got to spend a whole evening teaching Dr. Sylvia Earle about tardigrades. The first genus we found was by a student that had only been in my lab for 7 days. She found the specimen on the left, and then we got a beautiful SEM to confirm it on the right. Yeah. We take our students all over the world, India, Washington, D.C., young lady here is in a wheelchair, and she had climbed in the trees with us. Yeah. We promote bioindicators, the international transport of the animals with the bird feathers. That was a great discovery. It's really altered international distribution thinking. Yeah. We've explored the canopy in the last 10 years, partly because a student needed a project, and I said, well, what's in the top of that white oak tree there? And she climbed up and found more animals higher in the tree than at the base. So this has led to some of my students climbing some of the tallest sequoias in the world and collecting. And yes, we have tardigrades from the very top of these giants. We support student publication when the work meets peer-reviewed standards. We love to have collaborations this way. We've been lucky enough to have some NSF grants. We did a month-long tour in China, covered 10,000 miles, and had a great experience. We've done transcontinental in the United States, going to all the LTERs, and again, have added a lot of new data. The 2 REUs in the trees was an exquisite experience. If you ever get a chance to work with disabled folks with a scientific interest, they're marvelous. Okay, and we've met a lot of neat people along the way. Yeah. So our current project is trying to modernize the process of dealing with tardigrades. I realized that I teach students to collect and process and identify tardigrades exactly the same way I did 50 years ago. Oh, the optics have improved a little bit, and we have digital photography and that sort of thing. but we're not truly using modern technology to go after the animals. So we are looking to use tabletop robots to handle the slides. And then we're trying to use artificial intelligence to be able to let the computer identify the specimen on the slide from our image. Okay. We're also adopting drones, robotics to collect the animals out of the trees. In our climbing experience, we realized we were spending massive amount of time just rigging the tree. We could climb 2, maybe 3 trees a day, and it took hours to get the ropes up and get the ropes down and what have you. The actual collecting took an hour. By adapting a drone with a cutting tool on the front, we can fly up into many types of trees and collect and be gone in an hour, and do 5 and 8 trees a day. So it can also go across the stream and collect that tree over there, or pick off that rock that you can't get to otherwise. So there's a kind of exciting developments. We've explored advancing imagery, particularly with fluorescence, looking at pores here on the right in the green specimen, being able to see the plates and the distinctions between the plates that we can't see with the light microscope. Also right here, this little guy that has the white shoulder patch and the white cap, which are probably sunscreen protections. You cannot see it with the light microscope. And my dog just walked into the room too.
Well,
So
that may be the dog.
Huh?
Your dog may be timekeeping for us as possible.
Yeah. I'll be finished in just a moment here.
Okay,
Yeah.
thanks.
Here's some of the things we're looking at under the AI for trying to determine species. It seems what you can do with your eyes and your decision making should be computerizable. and so these are actual face contrast image level things that are quite easy to produce. And there's one of those eggs in a cuticle situation. And that's what it might look like if they're climbing around in a moss habitat, and be glad they don't get big. And that's the end.
Can you go back to the last one? That was so funny. That's okay. That's a great.
Previous.
The one before.
That one?
I wish I could hug a tardigrade.
Yeah.
So I have one question, because Annie does a lot of microscopy with mostly fleshy mushrooms, but also little bumps on logs and things. But we were talking before we got started tonight that she has never knowingly found a tardigrade. And so you talked about sort of the widespread distribution, but can you talk about the density? Like when you're taking those handful size samples, is there always going to be a tardigrade in that sample, or is it more hit and miss?
If you look long enough and far enough, you're going to find a tardigrade. To do comparison, you have to have some standard of comparison. And our subsample at 1 gram is pretty small. And in some cases, I can get hundreds out of 1 gram. And in about 30% of the cases, I don't find anything. If I reprocess the next one, I probably get 70% of those would be positive. So, and if you eventually went through almost every sample completely, which would take not only hours, but days and a massive amount of scope time, you would probably eventually find an animal. They're truly everywhere there's acceptable habitat. You find them up on the glaciers, in the cryo holes that are made by the birds flying over and pooping on the ice, and the sun warms the poop and it drills a hole in full of nutrients. And within weeks or 2, you start finding all sorts of nematodes, rotifers, tardigrades, what have you, in those cryo holes. Yeah.
Well, that's a great question,
Okay.
Elizabeth. And I'm going to start looking at lichen more now that I know that they're a favorite home of tardigrades.
Oh, they are. I've got bags sitting here. I get pulled over by airport inspection for having all these plastic bags of green leafy stuff.
I bet.
Yeah, it used to be a more of a problem. And we just use a handful and put it in the paper bag and wrap it up, and it'll sit there and wait for you for years. And I use ketchup cups out of McDonald's for processing. We weigh out the ounce and put in 20 milliliters of spring water from Quick Trip. Super simple to work. A pipette, a sample into. We use a little black Petri dish that's actually the lid to a plastic box, and we paint the back black, and it holds the 1 milliliter subsample that the pipette is. So everything gets standardized along the way. It works really, really nice for comparative data.
Oh,
Are you
cool.
soaking them in water to sort of float them off of whatever they're living on?
That's what the 24-hour floating is. You basically asphyxiate them, cause them to blow up by absorbing too much water. The animals that live in moss and lichen do not live underwater. They live in the interstitial space that contains water droplets like a sponge. And they're used to going in and out of cryptobiosis as that dries out and what have you. You think of a tree out in your yard, it may actually only be wet a few hours a year, soaking wet from rain, which is when the animals are active. The rest of the time they're cryptobiotic and dry, waiting. So those animals that live in that kind of condition, when you put them underwater all the time, they lose it and they go and float out, which is actually how we like them, because they are already stretched and they preserve real well and make nice slides. The marine ones never go cryptobiotic, and those that live in fresh water in streams and lakes never go cryptobiotic. And those are the two groups that we know the absolute least about, because they're harder to collect.
Interesting.
And so.
Wow.
Actually,
Well, I'm definitely gonna keep a much closer eye out in the future.
Anybody
Thank
that finds one, let me know. You can't
Yeah.
believe the number of cell phone images and videos I get asking me to identify the animal, which is almost impossible. We need a slide mounted specimen, of course, to identify.
So we'll hold on to it. If we find one, we'll hold on to it for you. Yeah.
I gave one of these lectures to the New York Microscopical Society a year or so ago. And a few weeks later, a guy sent me some videos and of a marine tardigrade. He lives out on Long Island, and we're in the process of describing a new subfamily, new genus, and new species of a tardigrade that has a beard. It's really obvious, and every time he walks around and he flips this beard up. And it's gorgeous. They also have different claws and what have you. He's a very distinctive animal.
Wow.
So we collaborate with people everywhere. I love to get small collections because I have finite time too. And we work with folks all over the world.
Well, I think you have some new tardigrade scats from DC, so we will definitely start looking. And thank you so much, Dr. Miller. That was a terrific talk and gorgeous images too.